Hello, I'm Maryam Shah, Research Assistant from Precision Medicine Lab. Today I'm going to talk about the dynamic protein interaction networks using fruit fly data to study cancer initiation and progression. So, what dynamic networks are? Dynamic networks are basically networks that change over time. With time, the number of nodes and edges change, and these networks allow us to look into persistent candidates throughout the time and to identify the featured nodes at each time course. As cancer is a complex disease and the molecular interactions in tumor change over time, therefore construction and analysis of dynamic molecular networks can provide a view to understand dynamic cellular mechanisms of different biological processes during cancer progression that could be helpful for therapeutic purposes. Due to unavailability of cancer temporal gene expression data, our study focused on dynamics of cancer gene orthologs to get insight about our cancerous genes. The study objectives were to construct dynamic network using protein-protein interaction and expression data of cancer gene orthologs, identifying persistent nodes in the networks over time, and also to perform functional annotation of the persistent proteins. The study also aimed to perform module and node centrality analysis of our dynamic network. Here we have shown the data source and methods we have used in this study. Cancer genes were identified from COSMIC database, while the orthologs and expression data has been obtained from Flybase. Then, the dynamic network was constructed and analyzed using a cytoscape app named DynetViewer, followed by finding persistent nodes using cytoscape. Finally, the functional annotation was performed using David Online Analysis tool. In this study, we identified 723 genes responsible for causing cancer. For these genes, we obtained 638 orthologs from Flybase, while the expression data was available for only 609 candidates. Using these datasets, we constructed a dynamic network consisting of 27 time courses. We identified 23 nodes that remained persistent over 27 time courses. Moreover, the dynamics of nodes and edges at different developmental stages indicate that both highest number of gene expression and lowest number of gene expression, represented by orange and red color in the table, are taking place during pupa development. We also performed functional annotation of persistent nodes that showed that these nodes are involved most prominently in biological processes that are cell shape regulation, centrosome duplication, phagocytosis, stress response, and cell adhesion. These nodes are also involved in some crucial molecular functions. These are kinase and protein binding, protein heterodimerization, transcription initiation factor activity. Moreover, in case of cellular components, results identified nucleosome, ribosome, centriole, nucleus, etc. After performing module analysis for our dynamic network, we obtained total 154 densely connected clusters over 27 time courses, where we can see that highest number of clusters are identified in time course 17. Moving towards the node centrality analysis, we identified 29 feature nodes with highest score of betweenness centrality, while 18 genes were obtained as feature genes based on highest degree centrality score throughout the total time courses. In this study, we identified 723 genes responsible for causing cancer. For these genes, we obtained 638 orthologs from Flybase, while the expression data was available for only 609 candidates. Using these datasets, we constructed a dynamic network consisting of 27 time courses. Continuing our work, we will further analyze the dynamic network to find out other essential features and will further study the feature nodes as well as the persistent nodes to look into their significance in the network. At the end, I would like to thank my teammates for their assistance and special thanks to our PI Dr. Faisal Khan. Thank you for your attention. For more information, you can email me on the mentioned email address. Thank you so much.